So very good afternoon, students. Today we are going to discuss about a fourth chapter, Principles and Process of Biotechnology. In the Principles and Process of Biotechnology, one of the important topic is the fermentation. So the fermentation we are going to discuss now. So the fermentation, that word derived from a Latin verb called fervor, which means to boil. Fervor, fermentation. So the basically derived from a Latin verb called fervor. Okay, fervor means what, sir? To boil. Kochidhan solrama illaya, to boil. Right. What is fermentation? Please note it down. See, while you are watching that video, please take your textbook. Okay, open the topic and underline with the highlighter. Objective questions as well as the important point should be properly noted in your textbook. Okay, whenever you open it, you would have seen that important points with the different colors. You can highlight with the points. Okay, use different colors for highlighting the points. Okay, now what is the fermentation? So the fermentation is nothing but it's a metabolic process. So this metabolic process in which organic molecules, especially there are so many organic molecules are there, protein, amino acids, okay, fat, etc., so many organic molecules, especially here the sugar, or you can say carbohydrate. So this organic molecule is converted into alcohol, gas and acids in the absence of oxygen and electron transport chain. So this process is known as fermentation. Okay, so you keep in your mind, fermentation is always takes place in the absence of oxygen. Underline that point. Underline. Look at your textbook and underline that point. It always takes place in the absence of oxygen, and not only in the absence of oxygen but also, and it's given the textbook. Look at the textbook. And electron transport chain. So in the absence of electron transport chain, so this process are usually occur. Okay, so this process called a fermentation. Okay, fine. Another important 2 mark or 3 mark questions given in the textbook. What is zymology? Underline, what is zymology? Zymology is nothing but to study fermentation and its practical uses, called zymology. To study about the fermentation and its practical uses are called zymology. Zymology nama solrom. Zymology. And this zymology, the term basically originated in 1856 from the French chemist Louis Pasteur. So Louis Pasteur demonstrated that fermentation was caused by yeast. Underline that word. The fermentation was caused by yeast. So yeast is responsible for the fermentation. So yeast will go anaerobic respiration in the absence of oxygen, in the absence of electron transport chain. It converts the sugar solution into carbon dioxide and alcohol. That's a fermentation. That's a fermentation. Okay. So that's why in 1856 a French chemist Louis Pasteur demonstrated that fermentation was caused by yeast. Underline yeast. Okay. So yeast contain one enzyme. Do you know? Anybody? Do you know? Okay. Or you tell me the name of enzyme which is present in the yeast. Nobody knows. Okay. So the yeast is contain enzyme called zymase. Z-Y-M-A-S-E. Zymase. Okay, so the zymase present in this. That's why to study about the fermentation and its practical uses are called zymology. Okay, right, fine. So, zymology, it's a branch of a science. Okay, there they are going to explain about the fermentation process. And basically, this fermentation was carried over by anaerobic organisms. There are some microorganisms, okay, as well as the fungi. Okay, they do not require oxygen for their survival. So they not depends upon the oxygen for survival. Such organisms are called anaerobic organisms. You know, anaerobiosis. Have you heard that word anaerobiosis? Anaerobiosis na life in the absence of oxygen. ஆக்சிஜன் அற்ற சூழ்நிலையில் உயிர் வாழக்கூடிய உயிரினங்களை தான் நம்ம anaerobiosis சொல்றோம். So these organisms, okay, bacteria and fungi, they can lead a life in the absence of oxygen. So they perform a process called fermentation. Okay, so the fermentation, the sugar solution is converted into alcohol and carbon dioxide. Right, fine. So fermentations are exclusively used in the food industries as well as the brewing and baking industries are exclusively using this fermentation process. Okay, and it is used for preserving of vegetables as well as, okay, and increase the taste and flavor in the milk products. Okay, so the acids which are obtaining from the fermentation that is used for to preserve the vegetables is not only used for to preserve the vegetable alone. Okay, it is also used to increase the flavor of milk products. Okay, and during the fermentation, you know very well, carbon dioxide is released. So that released the carbon dioxide gives the leavening of bread, softening of the bread. அந்த பிரெட்டில் உப்பு இருக்கும் பார்த்துருக்கீங்களா, அந்த உப்பு இருக்கிறது காரணம் கார்பன் டை ஆக்சைடு. அந்த ஆல்கஹாலிக் ஃபெர்மெண்டேஷன் நடக்கும்போது அந்த கார்பன் டை ஆக்சைடு வெளியில் வரும். அப்போ இந்த பிரெட்டெல்லாம் பார்த்தீங்கன்னா அப்படி உப்பி இருக்கும். ரொட்டிகள்லாம் உப்பி இருக்கிறதுக்கு காரணம் இந்த கார்பன் டை ஆக்சைடு. லீவனிங் ஆஃப் பிரெட்னா சாஃப்ட்னஸ் ஆஃப் த பிரெட். பிரெட்டெல்லாம் ரொம்ப சாஃப்டாக இருக்கும் பார்த்துருக்கீங்களா, ரொம்ப ஹார்டாக இருக்காது, சாஃப்டாக இருக்கும். காரணம் இஸ் கார்பன் டை ஆக்சைடு. ஓகே, ஸோ தட்ஸ் வை த ஆல்கஹால் இண்டஸ்ட்ரீஸ் and baking industries are exclusively using the process of fermentation. Okay, fine. Now this fermentation, how to obtain the fermentation product in a large scale? Okay, to get the fermentation product, we need to produce lot of acids. Okay, so we can obtain through the large scale by using this. One of the important instrument is called a bioreactor. Here you are looking at this is nothing but is a bioreactor, or otherwise called a fermenter. So what is a fermenter or bioreactor? Look at this fermenter or bioreactor. It is nothing but it is a big vessel, a very big vessel, okay, very big vessel used in the industries, and these vessels are modified such a way in order to provide okay, optimum environmental conditions. So this provide an optimum environmental conditions. Okay, so these optimum environmental conditions in which this microorganism, or sometimes you can use the enzymes. So this microorganism or enzyme can react with the substrate. Okay, they can react with the substrate and gives a product. So this product you can obtain in a large scale by using this fermenter. So fermenter or bioreactor, keep it remember, it always provide the optimal environmental conditions. Okay, so in the conditions we will get the product. Quality of product is depends upon what kind of this bioreactor fermenter you are using. Clear, right? So this look at this. Here we can mention some parts of this bioreactor. So bioreactor in which you can everything you are providing the controlled environment. If you want to adjust the pH, okay, easily you can adjust the pH because we are having pH probe. Look at this. This is a pH probe. These pH probes are used for, okay, to adjust the pH. And another one is called what to say there, temperature probe. the temperature probe used for to alter the temperature. We can provide a constant temperature to the fermenter, temperature probe. Okay, and these reactors are completely enclosed by cooling jacket. This is called a cooling jacket. Here, Tamil can English the cooling jacket. Okay, and also we can see the oxygen concentration probe here, because some microorganism it depends upon the oxygen. So we need to provide oxygen. So here we are having oxygen concentration probe. When you look at the textbook, they have mentioned in the textbook diagram. And here we are going to inoculate, nutrient or inoculant we can introduce in this here. Okay, and finally we are having motor. This one is a motor. Okay, and we can see a lot of the rotating fans, rotating blades. So this helps to mix the this culture medium with the enzymes. Okay, right. That's why this environment, suitable, optimum environment provide in which microorganism as well as the substrate, both they get together. They can react very well and produce the product. Okay, right. That's a bioreactor. So here controlled. What are the controlled conditions? They are providing aeration. Aeration is very important. Okay, aeration. We need to provide some purified air for the growth of this microorganism. Aeration followed by agitations. நம்ம கிளறிட்டே இருக்கணும்ல, நம்ம போய் உட்காந்து கிளற முடியாது. ஸோ அங்கே எஜிடேஷன்ஸ்க்கு ஒன்று இருக்கும், எஜிடேஷன்ஸ். அதுக்கப்புறம் டெம்பரேச்சர் அண்ட் pH all should be provided in a controlled condition. Okay, controlled. You are going to control everything. Clear? That's a bioreactor. So that's why I told you the bioreactor provide optimal environmental condition. Okay, in a controlled manner. That's a bioreactor. Okay. Now this bioreactor concept, two important processes are involved in the bioreactors. Okay, whatever the bioreactors it may be, it is involved in the two important main process. One is upstream process, another one is downstream process. Okay, what is upstream process? What is downstream process? In the upstream process, all the process before starting the fermentation. All the process before you starting the fermentation are called a upstream process. So in the upstream process, very very important, you need to prepare, and we need to provide a culture medium. So preparation should be done properly, as well as the culture medium should be sterilized properly. Sometimes the preparation as well as the growth of the culture medium should be sterilized properly. Okay, very important. So culture medium as well as the nutritive medium, whatever it will be, everything should be sterilized. That's why we need to prepare some inoculum. Okay, preparation as well as the growth of the culture medium sterilized properly before using that. Right, that is a upstream process. Okay, next one is downstream process. What is the downstream process? Okay, all the process after the fermentation are called a downstream process. All the process after the fermentation are called a downstream process. So this downstream process include distillation, centrifuging, and filtration, and extraction of the solvent. So these are methods are exclusively involved for this downstream process. The downstream process exclusively for the purification of the product. So how will you purify? You can purify the product because so many debris are there, unwanted substance will be there, in the product. So you want to remove all these undesirable, unwanted product from the product. Okay, so that's why we are using this method. One is called the distillation, followed by centrifuging, filtration, and this extraction of the solvent. So these methods are exclusively involved for this downstream process. So these are the two main processes are involved for bioreactor or fermenter. Okay, so I hope you that understand about this topic. So go through in the textbook. So the next next video will discuss some other important topic in the 4th chapter. Okay, thank you children.